Hi everyone, welcome to my YouTube channel. My name is Clement Chilambe from Clement Chilambe Tutorials, and welcome to the first tutorial in Fundamentals of Biomedical Sciences, focusing on the analytical techniques and lab methods, tutorial 1A. And this tutorial we are going to focus on the MCQs, multiple choice questions. So this is to just build on your knowledge and make sure that you are relating everything that we're going to discuss in this tutorial with the books that you've been reading and also the notes that have been given by lecturers, so that you can get most out of it. And make sure that you pause the video so that you test yourself before we shout out the answer actually. Okay, and if you enjoy my content, please like, subscribe, comment, and share the link to this tutorial. Okay, so the topic that we're going to focus on in our first analytical techniques and lab methods tutorial is the laboratory equipment and glassware. Okay, laboratory equipment and glassware. All right, let's get started. So we have our first question here. Okay, we have our first question here, which states a researcher needs to perform a series of acid digestions at 250 degrees Celsius. Which type of glassware would be the least suitable for this task? Okay, so make sure that you understand the question, especially the second part of the question. Which type of glassware would be the least suitable for this task? Okay, acid digestions at 250 degrees Celsius. A, borosilicate glass. B, soda lime glass. C, aluminosilicate glass. And D, quartz glass. Which one is the answer of your choice here, or which one is the correct answer here? All right, so the correct answer is soda lime glass. Okay, soda lime glass is less resistant to thermal stress compared to borosilicate glass, and may not really be suitable for high temperature or chemically aggressive applications. So the acid digestion at high temperatures, both a thermal and chemical stressor, so we need to take note of that. And this actually makes soda lime glass the poorest choice here. So the correct answer is soda lime glass. When we talk about borosilicate glass, the first choice here, this is explicitly noted for its excellent resistance to thermoshock, right? Even when you read through your books and also your slides, your lecture slides, you're going to note this difference here. And borosilicate glass actually is high resistant to chemical corrosion, making it ideal. And then when we talk of C here, aluminosilicate glass, this one offers good resistance to high temperatures thermoshock. Okay. How about D, quartz glass? Quartz glass is actually known for its exceptional thermal and chemical resistance, superior to borosilicate. Okay, so that makes soda lime glass our choice here to be the correct answer. Let's go to the second question. In a flow cytometry experiment, a scientist observes a significant increase in the side scatter parameter for a specific cell population. What is the most likely interpretation? Okay, remember there are two things that we're talking about here, two keywords. This is a flow cytometry experiment, and there is an increase in the side scatter parameter for a specific cell population. So which one is the most likely interpretation? A, the cells have increased in size. B, the cells have become more granular or internally complex. C, the cells are expressing a high level of a specific surface protein. And D, the cells are undergoing apoptosis. Which one is it's your answer here? Which one is the correct answer? You can pause the video and try to test your mind and answer the question. Okay, so the correct answer is B. The cells have become more granular or internally complex. Okay, remember the side scatter as a parameter measures the granularity or complexity of particles, and an increase in the side scatter parameter correlates with increased internal complexity. This is what we know. Okay, when we talk of the cells, option A here, the cells have increased in size. This one is measured by forward scatter, not really the side scatter. How about C? The cells are expressing a high level of a specific surface protein. For C, this would be detected by fluorescence from labeled antibody, not really by change in light scatter properties. No. How about D? The cells are undergoing apoptosis. Okay. Well, flow cytometry can detect apoptosis, right? This is what we know. but it typically does so through specific fluorescent markers, not really primarily through a standalone increase in the side scatter. So that makes choice B to be the correct answer for question number 2. Question number 3, which type of biosafety cabinet is characterized by having the operator work through attached gloves, provides the highest level of containment, and is used for agents like Ebola virus? Is it A, class 1 biosafety cabinet? Is it B, class 2 type A2 biosafety cabinet? Is it C, class 2 type B2 biosafety cabinet? Is it D, class 3 biosafety cabinet? Which one is your answer? So the correct answer here is D. Okay, so the correct answer here is D. Yeah, because we know that class 3 biosafety cabinet offers the highest level of containment, and that personnel work through gloves attached to the cabinet as keywords in our question. So we agree with this as well. And it's actually it's class 3 biosafety cabinet is specifically mentioned for handling high risk agents like Ebola, including SARS-CoV-2 as well. Okay, so our answer for question number 3 is D. For class 1, remember, class 1 provides personnel and environment protection, not really product protection, and does not involve glove points as well. Then for B and C, these are the most common types and provides protection for personal, product, and the environment, but they are not gas tight or used for the highest risk agents like class 3 cabinets. So D is the correct answer. Question number 4. A volumetric pipette is best distinguished from a mole pipette by, okay, so the way you're pronouncing pipette or pipette, I don't know, but it's a pipette, really. A volumetric pipette is best distinguished from a mole pipette by A, its ability to measure variable volumes, B, the presence of a single specific volume marking, C, graduations that extend down to the tip, and D, being made of borosilicate glass. Which one is the correct answer here on question number 4? So the correct answer is B, the presence of a single specific volume marking. Okay, why is B the correct answer? Because volumetric pipettes are actually designed for a single specific volume. Okay, but when you talk of graduated pipettes, and an example of a graduated pipette is the mole pipette. These have markings for different volumes. Okay, so when you talk of now option A is ability to measure volume variable volumes, this one is for the graduated pipette. And when you talk of C, graduations that extend down to the tip, this one is for say a serological pipette, right? And then for D, being made of borosilicate glass, the material is not really the defining feature here, and because both types can be made from the same glass, so this is not our focus. So our focus is actually B. Yeah. So question number 5. Let's go to question number 5. The primary function of the coarse and fine adjustment knobs on a combined microscope is to do what? A, to control the intensity of the light source. B, to what's B? Adjust the focus by moving the stage or objectives. C, change the magnification of the objective lenses. And D, adjust the diameter of the aperture diaphragm. So in laboratory equipment and glassware, the lecture is incomplete without mentioning of microscope. It's actually a fundamental tool in biomedical sciences that helps us in diagnosis of the sample that we are working on, right? In microbiology, in parasitology, even in histopathology, even in hematology, a microscope is an indispensable tool, right? So now let's answer our question. So which one is the correct answer here among the 4 options that we have? So among the options that we have, we go for option B, adjust the focus by moving the stage objectives. Of course, you need to know the features of a microscope. You need to gain some skills that will help you ensure that you become a good laboratory personnel and a good biomedical scientist with the way you are working with the microscope. But yeah, it's also important to note that there are some features which are very important to know, especially in the diagnosis itself. And one of those features are the coarse and the fine adjustment knobs, the knobs rather. So why are we saying our correct answer here is B? a standard microscope, these two features here, which the parts rather, the coarse adjustment and the fine adjustment, they are the standard microscope controls for focusing the image by altering the distance between the objective lens and the specimen. Why is A not the correct answer? Okay, the controlling the intensity of the light source. This is done by the brightness adjustment, right? Or the voltage control of the illuminator, not these, the coarse and fine adjustment knobs. Then C, why is C not really the correct answer? Change the magnification of the objective lenses. This is done by rotating the nose piece to select different objective lenses, right? So not for coarse and fine adjustment knobs. D, why is it not the correct answer? This is adjust the diameter of the aperture diaphragm. This is actually a separate control, and it's often located beneath the stage. It is used to control the contrast and the depth of a field, right? So our correct answer remains undefeated. Okay, so adjust the focus by moving the stage. So now we move to the next question. Question number 6. In the context of flow cytometry is back again. What is the purpose of hydrodynamic focusing? A, to excite the fluorophores attached to the cells. B, to ensure cells pass through the laser beam one at a time. C, to sort cells into different collection tubes. D, to filter out debris and cell clumps from the sample. Which one is the correct answer? Our correct answer is B, to ensure cells pass through the laser beam one at a time, right? You know, when we talk of the flow cytometry, the sample is hydrodynamically focused into a single file stream of cells, right? And that ensures that each cell passes through the laser beam individually. And this is very, very important for the accurate measurement of each cell's properties, okay? And looking at option number 8, to excite the fluorophores, this is actually the function of the laser beam itself. Then why is C not the answer? This C here, to sort cells into different collection tubes. Okay, so this is a function of specialized flow cytometers called cell sorters, right, which use different mechanisms. By different mechanisms, I mean, for example, what example can I give? Electrostatic charging. Okay, so these cell sorters use different mechanisms after analysis, so not really our concern. Then D, why is D not the correct answer? To filter out debris and cell clumps from the sample. Okay, so to filter out debris, this one is done prior to injection to the flow cytometer to remove clumps. Okay, yeah. Question number 7. A laboratory is processing tissue samples for histopathology. After fixation and dehydration, the tissue must be infiltrated with paraffin. Which piece of equipment is specifically designed to automate this multi-step process? Okay, yeah. You know, when we talk of our diagnostic laboratory, the setup is that many have got the histopathology section. This is where we ensure that there are a lot of things that happen in histopathology, and there are stages of things from sample reception to the processes such as sorting, grossing, of course, and we pass the tissues through so many processes until we have the final view that we need. Okay, this is where we analyze cells to see the abnormal cells, just using these simple terms, right? Let me just end there. And which one do you think is our correct answer here? Which piece of equipment do we use in histopathology lab when it comes to tissue sample processing, according to the scenario that we've been given, rather? Yeah. So among the many pieces of equipment that we use in histopathology, but for this specific one, we use the automatic tissue processor. Okay, we use the automatic tissue processor. So the automatic tissue processor, it is used to automatically prepare tissue samples by fixing, dehydrating, clearing, and also infiltrating them with a paraffin. Yeah. Why is A autoclave? When we talk of autoclave, autoclave is used for sterilization using steam and pressure, not for tissue processing. Microtome is also a piece of equipment in histopathology, but it is actually used to slice the paraffin embedded tissue into thin sections after processing. What about biosafety cabinet? Biosafety cabinet is actually used to provide a sterile and contained environment for handling hazardous materials, not really for tissue processing steps themselves. All right. Question number 8. Why is it critical to work with a sash of a class 2 biosafety cabinet in the proper position? Yeah, typically not down to a specified height. This is hard to put this in brackets because, yeah, this is to make you know, to let you know that this is how it should be done. All right, so A, to improve the ergonomics for the user, B, to maintain the correct airflow pattern and ensure containment, and then C, to protect the samples from airborne contamination from the room, and D, to reduce the noise level from the cabinet's motor. Okay, now correct answer here remains, or maybe not remains, but rather to maintain the correct airflow pattern and ensure containment. B, without question, really, because yeah, the regulations are that we need to always work with the cabinet sash down to maintain containment. This is class 2 biosafety cabinet. The sash height is calibrated to ensure the inward airflow or the air curtain is sufficient to protect the user and prevent particles from escaping. Yeah. Why is A not really the correct answer? To improve the ergonomics for the user. Is this, well, this is actually a side effect, but it's not really the primary safety reason that we are considering here. Then C, to protect the samples from airborne contamination from the room. Okay, yes, class 2 biosafety cabinets do provide product protection, but the primary reason for the sash is personnel environmental protection containment. Yeah. Then G, to reduce noise. This is not really the function of the sash, really. So the correct answer remains B. A micropipette is mostly appropriate use for measuring which of the following volumes. This is nice and not really tricky. I wouldn't say tricky, right? So a micropipette is mostly appropriate used for measuring which one of the following volumes. A 750 microliter, B is 25 microliter, C a 150 microliter, and D a 500 milliliter. Did I say on B 25 microliter? It's actually milliliter. Hope you are seeing this right. Now, which one is the best answer here? Okay, so now okay, let me just state the answer, then we're going to state the reasons as to why it's the correct answer and why other options that we have here are not really the correct answer. So our correct answer here is C, 150 microliter. So now here are the reasons why. When we talk of 150 microliter, of course, let's first look at the micropipette, right? So micropipettes are designed for very small volumes, microliters, right? Which qualifies our answer. And then both 150 microliter and 750 microliters are in the microliter range. Of course, still qualifying our answer. But the problem with A, the 700 microliter here, this typically is typically measured with a larger pipette, right? And often a 1,000 microliter micropipette, yes. So yeah, that's why C qualifies here. Okay, 150 microliter is a standard volume for a micropipette. Okay, so the key is that micropipette handles microliters, not milliliter, right? And then of course we've already said under the 750 microliter, of course it is the microliter range, but this is a high volume for a micropipette and might be better suitable for suited for a serological pipette depending on the required precision. Yeah, 25 milliliter and 5 milliliter, these are milliliter volumes which are measured using larger pipettes or graduated cylinders, not really micropipettes. Yeah. Number 10, the resolution of a microscope is A, the ratio of the image size to the objective size, B, the ability to maintain focus when switching objectives, C, the smallest distance at which 2 points can be distinguished as separate, and D, the total magnification power of the objective and ocular lenses. Which one do you think is the correct answer here? The resolution of a microscope is what? Okay, so the correct answer here is the resolution of a microscope is the smallest distance at which 2 points can be distinguished as separate. C. All right. So why is A not really the answer? This is saying the ratio of the image size to the object size. This is not resolution, this is magnification. Then C is saying the ability to maintain focus. Okay, when you talk of the ability to maintain focus, this describes something that we call in laboratory lab science as perfocality, right? Which is actually a feature of some microscopes. Then D, the total magnification power of the objective and ocular lens. This one, this is a calculation. This one is a calculation. The objective lens multiplied by ocular lens, not really a definition of a resolution. Yeah. In a PCR experiment, what is the primary function of the device? That is not explicitly detailed but is central to the process of amplifying DNA. A, to separate DNA fragments by size. B, to sequence the amplified DNA product. C, to perform cyclic temperature changes for denaturation, annealing, and extension. D, to measure the fluorescence of the DNA samples. Which one do you think is the correct answer here? Okay, so the correct answer here, please at some point pause the question so that you can answer, test your mind, test yourself if you really know the answer or not. Don't be passive, be active during this tutorial. Okay, so which one is the correct answer here? To perform cyclic temperature changes for denaturing, annealing, and extension. So C is our correct answer, right? So we're talking about the polymerase chain reaction here. So, of course, a comprehensive understanding of laboratory equipment requires knowing that a thermocycler is actually the essential instrument that automates the temperature cycles required for a PCR, right? But now, look at A. Look at A to separate DNA fragments by size. This is done by electrophoresis, right? This is done by electrophoresis. Then talk of B to sequence the amplified DNA product. This one requires a DNA sequencer, which is actually a different instrument. And then when you talk of D, to measure the fluorescence of the DNA samples. Okay, so to measure the fluorescence of the DNA samples, D, this is done in quantitative PCR, right? It's quantitative PCR machines. These quantitative PCR machines are a type of a thermocycler with detection capabilities, right? But the core function of any PCR machine is thermocycling. Yeah. Our answer still remains undefeated: to perform cyclic temperature changes for denaturation, annealing, and extension. Yes. 12. What is the critical safety feature of a hot air oven that prevents heat loss ensures even controlled temperatures? A, asbestos door gasket. B, main on and off switch. C, direct through timer. Then D, tray for sample. Which one do you think is the correct answer here? Make sure that you understand the question. What is the critical safety feature of a hot air oven that prevents heat loss and ensures even controlled temperatures? So the correct answer here is asbestos door gasket. What did I want to say? I wanted to say asbestos asbestos door gasket. All right, so the asbestos door gasket is the correct answer, because actually the function of the gasket is to create an airtight seal on the door, which is essential for preventing heat loss and maintaining a uniform internal temperature. And modern ovens use non-asbestos gasket materials due to health concerns. This you should know. And why should we say main on off switch is incorrect? Yes, because this controls power but does not really contribute to temperature uniformity, right? Then direct through timer, this controls the duration of the operation. Then tray for sample, this holds the contents but does not really insulate the chamber. So our answer remains undefeated. Asbestos door gasket is the correct answer. A laboratory needs to sterilize a large volume of liquid culture media in glass bottles. Which type of autoclave would be most suitable? A, pressure cooker type. B, vertical autoclave. C, horizontal autoclave. D, large automatic hospital autoclave. Which one is the correct answer here? Now, you can pose the question. and think through the answer. I mean, think through, you know, ensure that you understand the question. You get the your choice here among the four choices that we have, so that even if I've switched out the answer, you're able to relate and you're able to clap for yourself because you have got the correct answer out there. Okay, so the answer is horizontal autoclave. Here, C, horizontal autoclave. Remember, a laboratory needs to sterilize a large volume of liquid culture media in glass bottles. Which type of autoclave would be more suitable? Horizontal autoclave. Why? Horizontal autoclaves typically have a cylindrical chamber. Okay, these cylindrical chambers lies horizontally, and a door that opens from the front, making them ideal for loading large or heavy items like stacks of bottles on trolleys. Yeah, in trolley, you know, we have a picture of that. Now, the large automatic hospital autoclave, which is our D option here, could also work, but horizontal autoclave is a standard type for this purpose, right? The pressure cooker type, this is a small benchtop unit and suitable for large volumes. And then the vertical autoclave, these are small, these are more compact, right? These are more compact and are loaded from the top, which can be, you know, cumbersome for large heavy bottles. Yeah, and for large automatic hospital autoclave, well, potentially suitable, but the horizontal type is the classic design for this application lab context. 14. In a flow cytometry, forward scatter is a direct measure of a cell's A, granularity, B, fluorescence intensity, C, size, D, viability. Which one is the correct answer here? In flow cytometry, forward scatter is a direct measure of a cell's what? Which one is the answer? All right, so the answer is size, right? The answer is size. Yes, forward scatter measures size of particles. Yeah, granularity, this is measured by the side scatter. Then fluorescence intensity, this is measured by fluorescence detectors after a cell is labeled with the fluorochrome, isn't it? Yes. Then viability, this can be assessed using a flow cytometry with a specific fluorescent dyes, but it is not what forward scatter is directly measures here. So our answer remains undefeated. Size. Okay, question number 15. The part of the micropipette that is responsible for securely holding the disposable plastic tip is called the A, the plunger, B, the barrel, the C, the tip holder or tip cone. I feel like I'm doing forensic pathology here. The barrel. Now D, ejector button. All right, like a gun. Okay, I'm not commenting anything. But which one is our correct answer here? The part of micropipette that is responsible for securely holding the disposable plastic tip is called the tip holder or tip cone. C is our correct answer. Okay. The tip holder or tip cone, what can I say about this? It's actually the conical end of the pipette shaft onto which the disposable tip is pressed to form an airtight seal. Yeah. Why is the plunger not really a suitable answer? This is used to aspirate and dispense liquid, remember. Then B, the barrel. This is the main body of the pipette that houses the mechanism, the whole mechanism. And then the ejector button. This is used to remove the tip after use. So our answer remains undefeated. The tip holder and tip cone. Number 16 rather. A class 1 biosafety cabinet is characterized by A, providing both personnel and product protection with HEPA filtered recirculating air. Then B, being a gas a tight enclosure with glove ports for the highest level of containment. C, providing personal and environmental protection by exhausting HEPA filtered air. And D, having an open front and relying on laminar flow to protect the product. Which one is our correct answer here? Okay, so B, no, not B. Are you saying B? C. Okay, providing personnel and environmental protection by exhausting the HEPA filtered air. Okay, yeah. So a class 1 biosafety cabinet provides personnel protection by drawing room air in and exhausting it through the filter, the HEPA filter, and this one explicitly states no product protection. I think that's my first clue when it comes to class 1 biosafety cabinet. Whenever I see or whenever I remember that there's no product protection, then it's easier for me to even find the answer here among the choices that have been given. So why is A not really the correct answer? When you look at A closely, it provides both personal and product protection. This describes a class 2 biosafety cabinet. Then B, this one describes a class 3 biosafety cabinet. Yeah, gas tight enclosure with glove box. Then D, having an open front. This describes, yeah, this is easy. This describes a laminar flow hood or a clean bench, right? Which protects the product but but offers no personnel protection. Yeah. The component of a spectrophotometer that is responsible for separating light into its constituent wavelengths is the A, the cuvette, B, detector, C, monochromator, D, light source. Which one is the correct answer here? Of course, we've even entered the other lecture, but yeah, since the spectrophotometer is one of also the indispensable tool or equipment that is found in the lab, of course, let's discuss this question. Now, which one do you think is the correct answer here? Which part of the spectrophotometer component is responsible for separating light into its constituent wavelength? The correct answer is monochromator, right? Yeah. When you want to understand something about a spectrophotometer as a method or as a technique in the laboratory, you need to know that its principle requires knowing that a monochromator, of course, when you talk of a monochromator, it can include a prism or a diffraction grating, right? So a monochromator is a component that selects a specific wavelength of light to pass through the sample. A covariate here this is just the container that holds the sample, right? The detector just measures the intensity of the light that passes through the sample, and the light source just generates the broad spectrum light. Okay, it can be the deuterium lamp or it can be the tungsten lamp. Yeah. 19. We go to question number 19. What's the fundamental principle that allows a burette to deliver variable volumes of liquid with high precision? A, it is calibrated to contain a specific volume. B, it has a stopcock for a controlled delivery and fine graduations for accurate reading. C, it is made of high quality borosilicate glass to prevent chemical reaction. And D, it has a tapered tip to allow for slow drop by drop addition. Which one do you think is the correct answer here? What is a fundamental principle that allows a burette to deliver variable volumes of liquid with high precision? We all know what a burette is. And if we really do, then we are going to consider B to be our correct answer. Yes. So the combination of the precisely engineered stopcock for flow control and the fine graduations along its length is really what allows for the delivery of variable precise volumes. In case you're asking yourself, how does the burette deliver variable volumes of liquid with high precision? Yeah, the precise engineering of the stopcock. Now, it is saying it is calibrated to contain a specific volume. And, this describes a volumetric flask. Okay, is a volumetric flask. C saying it's made of high quality borosilicate glass to prevent chemical reaction. This is a feature for safety and durability, not the principal volume of delivery. Okay, then D, it has a taper tip to allow for slow drop by drop addition. This one, of course, this aids in delivery, but it's not really the defining feature for precision. And you need to take note, of course, that a graduated cylinder also has a taper tip, but is less precise. Yeah. The use of standardized laboratory glassware and equipment directly supports which of the following scientific principles? Actually, I thought this was supposed to be the first question among the 20 questions that we have, and this is our last question. This was supposed to actually be our first question. So now let's make it nice. Let's make it interesting. The use of standardized laboratory glassware and equipment directly supports which of the following scientific principles? A, precision and accuracy only. B, safety and specialized functions only. C, reproducibility and precision. And D, all of the above. The answer is all of the above. Yes. The 4 key importances, right, is what you need to know about the laboratory glassware and the equipments rather. So precision and accuracy, safety, reproducibility, and specialized functions. Actually the key importances of the laboratory glassware and equipment, right? Standardized equipment is fundamental to achieving all of this, yeah. So A, B, and C are all partially correct, but do not encompass the full scope of benefits, the benefits that we might need for the complete answer on question number 20. So this was our last question, right? So thank you so much for staying with me throughout the entire tutorial. Ensure that where you do not understand, you go back, you play the video, you enjoy the explanations and also the answer choices that we are discussing throughout the tutorial. Of course, this is Clement Chilambe Tutorials, and subscribe, like, and comment if you do like enjoying my content. Of course you do. Make sure you do that. You share, you make sure that you share, and you invite others to also subscribe to the channel. Thank you so much. Until next time, see you. Bye.